Morning, everybody. Welcome back to the Exito URISE Annual Symposium. For many of you were with us on Tuesday, so you know how this goes, but I'll just give a real brief introduction. First of all, we're going to have presentations just for 2 hours until 11 o'clock, and then we shift over to the lobby for the poster session. So we have several presentations that will be 5 minutes each, and then 5 minutes for question and answer, and then a little time in between for the transition. So again, we've got that 5 minutes in between, so we're not going to keep you strictly to 5 minutes if there's a good discussion going, but we'll keep things moving along because this is posted for people who are joining us by Zoom, so they're trying to log in to see certain presentations. So we want to stick to the schedule. Just have a couple other announcements. First of all, again, this is the kickoff for the new school year. So that means enrichment is starting up again. And the enrichment team is encouraging scholars from all cohorts to attend next Friday is that tomorrow or? Sure. Maybe tomorrow, maybe Friday next week, from 1 to 2:30. And the point of that session will be to sort of debrief the symposium and talk about the experience of presenting and doing posters and everything else you did this summer. So we want people, students and scholars from all cohorts to talk about their research experiences this past summer. And it's sort of a structured networking between all of the cohorts as well. So we want all cohorts each program year to be involved in that. So if you have any questions about the date, like I do, check the enrichment website, and I'm sure the details will be there. So anything else before we get started? Are we on schedule? There you go. All right. Well, it's my pleasure to introduce our first presenter for today, Jacqueline Garcia, and about research. Thank you. Oh, I'll just mention that in this room, all of the presenters are from the URISE program, both cohort 1 and cohort 2. So cohort 1 has had 2 summers of research plus last school year. Cohort 2 is just entering their research placement. So they're talking about things they've just started on this past summer.
Hello, my name is Jacqueline. This is my first summer with Dr. Hinson at the Division of Trauma, Critical Care, and Acute Surgery at OHSU. The project that I have been tasked with being a part of is predictors of low-risk phenotypes after traumatic brain injury, incorporating proteomic biomarker signature. In the lab space, we just call it ProTips, which is much easier. So I might call it ProTips further on in the presentation. Okay, introduction, traumatic brain injuries, acronym TBI, are a leading cause of death. There are limited treatment options. And so according to the CDC, the Center of Disease Control, TBI-related deaths in 2020 was 64,362. First, this might not seem like a lot, but then there are cases that aren't reported in this number, which are those that are only treated in the emergency department, primary care, urgent care, or those that go untreated, as is the case with a lot of things, right? Okay, so what is TBI? It's an acquired injury to the brain. Most of the time when someone says TBI, you think, oh, concussion, which yes, it falls under TBI and it's a result from trauma. And among civilians, the most common cause would be falls, especially among the elderly. Accidents, anything really with a moving vehicle, bicycle, motorcycle, car. Okay, and so this, I won't read much off of the image, but it's the Glasgow Coma Scale called GCS. And in this one, this is what we use to measure the severity of TBIs. And so it's a scale from 3 to 15. 3 to 7 would be severe. This could fall unconscious, admitted to the ICU, intensive care unit, could be connected to breathing tube. So very severe. And moderate, that's 8 to 12. That could be not fully there, but still kind of okay. You could get admitted to the hospital. Mild 13 is a score of 13 to 15. This one, I would say all of us are GCS of 15. We're here, we're good. We're very oriented. We can hear, we're good. We can respond, move, open eyes. So what are we studying? So our objective is try to create a statistical model to predict patient outcomes. This includes clinical data, which is just like vitals and graphics, head CT scans to see the progression of bleeding in the brain, and also the protein from the blood, which is a vial we get within 3 hours of the TBI. And the group we are enrolling now, it's observational perspective. Observational meaning we're only observing, we are not changing anything in the way they're being treated. We're not giving them vaccines, injections, anything like that. Perspective being it's in real time, what is currently happening. We aren't looking at past medical records, it's just what's happening at the moment. We're still on the enrolling patients into this study. So the goal was 126 within 3 years. We are currently at 102. That's very close. I did mention previously that it's statistical outcomes. These two, the short-term outcome is measured by the progression of bleeding on the CT scan with the 6-hour difference between those. And then long-term would be the neurological function at 6 months using the GOS, which is similar to the Glasgow Coma Scale, but it's the Glasgow Outcome Scale, which is usually followed up 6 months after they've been released, and it's through a phone interview. Summary, why is this research important? We're hoping that this model will help doctors be able to give better treatment options. So if we're able to tell right when they get admitted, if they're going to have a short visit or a more longer visit, we could be able to give better treatment options. And if looking at the blood proteins from the sample of blood that was collected originally, after we determine how the patients will, what the patient outcome will be, we might be able to see if some of them can lead us to say, okay, since they have this particular protein more of, then they're most likely to have this particular outcome. And acknowledgements, you rise clearly. Dr. Hinson, who's my RLC, and the Trauma Research Lab at OHSU. And then I also included the grant numbers. Thank you for listening. I know it was very simple, but questions? Yes.
So there are some settings that you said that TBI deaths go unreported, like the ER and the urgent care. Could you expand on that and why they go unreported there?
I wouldn't be sure, since all I know is really that the Center of Disease Control is the one who gathered the statistics. Might be that just didn't have the time, or I mean, this is just speculation on me. I'm trying to think also why that would be the case. It might just be because it's not a long enough stay or something like that, because there are cases, right, when you get someone and they might be released like maybe 2 or 3 hours after. So, but that would probably be my speculation, but good question. Yes.
Could you tell us a little bit about what proteins you're looking for that might correlate with BT?
Yes, I don't remember their abbreviations, so I do know it's 2 particular ones, if I'm not mistaken, that we're looking at. One is specifically to the short-term outcome, and another one is to the long-term outcome. Though I do have a file on my computer with that information. If I can share that if you'd like.
Thank you.
Yes.
Are there any studies going on in your lab currently that look at the cultural differences that might make someone more likely to get follow-up care for a TBI versus someone who just goes to the emergency department once and might have like critical need?
I wouldn't know. I don't want to answer that incorrectly, so I'm going to say that I don't know, honestly. I do know that there's some other ones, other projects going on, but the main ones that I've been listed to are ones that kind of look at statistical data and clinical data rather than visits like that. But I will ask about that to see, because now I'm curious as well.
A couple. So you sort of mentioned that maybe elderly people are more prone to some of these, and then I would imagine that younger people being less risk averse might also maybe have a higher prevalence. Can you maybe speak to like what the population looks like a little bit more?
Oh, in TBI?
Yeah, and then I guess I have a second question on the 15 scale. I think just like you said, we're in that 13 to 15 range, and like I might miss one because I didn't have coffee in the morning or something. Or elderly people also like maybe just inherently scoring a little lower on that scale.
Yeah, so there is a team dedicated to deciding on the GCS score, so they are trained in that, so just not just anyone can give out that score. I have been given the chance to shadow, and they are right, it's a lot of elderly people who come in because of falls. But I don't know a lot on the statistics because it's not really something I have looked for. Can you repeat the first question? I think I might have crossed
That was
over it.
basically it, yeah, and just like how that might affect biomarkers if your population is sort of like in, it's like bi-directional, it's not.
Oh, yeah, so there is as particular, so when we're looking for patients to enroll, we're hoping that they are 14 years or older, also with a GCF score of 12 or lower. And then obviously, since we need the sample of blood bio, we need to get that within 3 hours of when they had the injury. And sometimes they come in later, so we're not able to get that even if they fill in everything else. And then they also have to meet having the bleeding in the brain when we take the CT scan. So in most cases, when they get a score of usually when that's asked for. Did I answer your question?
Yeah,
I'm not sure.
that.
Okay.
Thank you, good talk.
Yeah, thank you. Yes.
How injury severity is taken into account, like someone versus like someone who like had a fall off the ladder or someone who was hit by a car, those are gonna be like different types of TBIs. Are those grouped? Or are they like, is there a difference too?
So as far as I know, we use the GCS to measure that. So from what I've seen, usually on falls, it gets a GCS score of 12. And I think that's the one I've most been. And then on car accidents, it really just depends on like the speed of the car and like what you're in. For example, if it's a bicyclist or another car, that's where the GCS tends to differ. Just, yes. Okay, thank you. Yes.
Really nice talk. I was curious of how often the GCS is assessed when someone comes in for a DI.
Okay, so we're looking at vitals 48 hours after admission. So we're hoping to get an hourly of that. So that includes vitals such as mean arterial pressure, temperature, and the GCS. So we hope to get an hourly on that. You? Yeah. I don't think I missed anyone. Thank you for listening.
All right, we'll move on to our second presentation. So I'm pleased to introduce Megan Creech, who's going to be talking about research.
This is a preliminary analysis of spindle density in participants that use cannabis frequently or in the study. And I'd like to thank the OHSU CPCP lab for inviting me in and welcoming me into their research process. Let's talk about sleep. Over here, you're awake, you're alert. We just talked about this. Here, you're dreaming. Here, you're a little bit asleep. You're probably denying it if anybody wakes you up. This is what we're looking at. Here, you're real deep. I don't care. This is the sleep stage that I looked at in round 2. It is the longest sleep stage, and it's where sleep spindles occur. What is a sleep spindle? It's one of these. It's real hard to see them in high res. They're very ugly. Sleep spindles are a measure of cortical activation in the brain. We use leads that have to cross the brain to pick up the electrical activity. And they've been correlated with memory consolidation and learning, as well as general cognition. Higher sleep spindle density has been correlated with greater cognitive performance. So sleep spindle density is a measure of how many spindles you're having per minute of NREM sleep. I guess. Why we should study this? Individuals with long-term cannabis use show cognitive deficits in midlife, specifically in hippocampal density. Both THC exposure and sleep deficiency are associated with attenuated cognitive function. So both people who have imbibed and people who are suffering from sleep deficits tend to test poorly on cognitive tests. Sleep promotion is one of the effects sought by individuals seeking out cannabis. So people are going to look for cannabis to provide sleep aids. This is the synthetic Delta-9 THC that we use because cannabis is still federally illegal to study, at least in its naturalistic applications. We hypothesized that participants with frequent cannabis use would have significantly lower sleep spindle density than their cannabis-naive counterparts. And then these are our specific participant class definitions. Frequent cannabis use was defined as over 3 uses per week with a positive urinalysis, which means positive for THC metabolites. Cannabis naive meant that they had fewer than 10 lifetime exposures and their urinalysis was negative for THC metabolites. And this is the timeline of the experiment. So participant arrives midday, and then right before they go to sleep, a placebo is administered on the acclimation night. They sleep. In the morning, they undergo a battery of cognitive tests. And then this is their daytime. Another placebo is administered. This is the baseline night, because this night for everyone in the sleep lab is just, oh no, I'm sleeping in a new place and everything's weird. So this is when you can actually measure what their sleep tends to look like on a regular day. This, in the green outline, is the drug exposure night. So this is when, instead of being dosed with a placebo, they were dosed with pronabinol. And then in the morning, they did another battery of cognitive tests, and then they left. And this is when I showed. And this is where I had to get help. So a criteria was developed for what a sleep spindle was. We chose to only look at spindles that were of a duration between half a second and a second and a half, with amplitudes between 0 and 60 microvolts and a frequency of 11 to 15 hertz, though both of those are pretty hard to look at. This is what the software that you look at sleep spindles on looks like. It's a PSG file. And each sleep spindle has to be hand highlighted. So this is still a human scoring process. There are machine automated processes, but they actually are still not as reliable as humans. And then these are our preliminary results. So we did not prove our hypothesis. Shockingly, 3.8 plus or minus the standard deviation of 1.3 versus 3.8 plus or minus the standard deviation of 1.2. So my supervisor said that he doesn't see these ever. This doesn't happen. This is highly unlikely. Future corrections are to have another build exo scorer come in. Hi, sleep spindle scorer number 2, and to increase inter-rater reliability. I scored sleep period 1 and sleep period 2, and Shidell will score sleep period 2 and sleep period 3. These are my references. These are my acknowledgements, of which I have a lot. Thank you, everyone on the planet, pretty much, but specifically the CPCP lab. I want to thank Dr. Chandy Dixon and Decatur Foster, and I also want to thank Dr. Bowles and Dr. Gonzalez. Yeah, thank you to the School of Public Health. Do you have any questions?
The counts over the 3 separate days, are they identical over the span of the 3 days, or do they differ at all?
The cognitive tests were the same.
Okay, and what kind of cognitive tests? Were they?
One was a word recall test. Another one was a psychomotor vigilance test. And I think there was a Passat test. Because I was only able to look at the first 2 nights, the cognitive tests were not like super relevant to the part of the research that I was doing, but I did look into them. And standardizing them and then testing the same person across multiple nights actually increases the analysis power of the study. It makes our results more reliable to be able to get that many results from one person and then repeat it in multiple people.
Is there carryover from your treatment, your THC night, and is that why you didn't randomize the order of the placebo versus the treatment?
Can I ask you to clarify your question about carryover?
Yeah, so if you gave them so a lot of times in designs where you have a placebo and a treatment, you randomize the order so that it's not that they got one first and that's what's how you see your difference. So I'm wondering if the reason you had to do it the third night was because there could have been carryover to the fourth night. So doing it first would affect your placebo.
I think that I can speak to that in terms of like the limitations of the study. Part of the limitations are that when we see here, I'll go back to my experimental timeline. So because everyone in a sleep lab will have an activation night. And then because we had 2 participant classes, one class was frequent users or people with frequent use cases. The frequent use cases had already been asked to abstain for 72 hours prior to the experiment. And then in addition, the activation and baseline nights, they were not using because they were in the sleep lab. So there is a little bit of funniness around how acute their use actually is, since there's almost a week since they have last survived. But I really can't speak to the randomizing of nights because I'm still so new to sleep science. But if you want to email me, I can definitely get clarification.
I was just curious, thanks.
What was the criteria for your test subjects, and was there any subjects that you didn't study, like patients with sleep apnea or anything like that?
Sure. Yes. So this study was done specifically on, I'm gonna put this in quotes just because I'm disabled, healthy participants. So that meant that chronic illnesses were ruled out. They were also given a psychological test to make sure that this was not going to be exceptionally disturbing to them. And then they were, tested for apnea, and during scoring of the spindles, there were certain conditions during a specific night that could affect whether or not those spindles got counted. Like if that person just snored, then the spindles around that are going to be a little suspect. Does that answer the question?
Yeah, thank you.
I was really,
Oh, sorry. I don't know, there was pointing at me.
I was really interested in the comment they made about cannabis being legal federally and how that impacts the research. I was interested in if you think that like impacts the results that you're putting out to people that are actually consuming cannabis instead of like something synthetic.
My personal opinion is that yes, it's very much a factor. I think that this is one of the ways that we are currently allowed to look at this science. But I do think that I personally believe that when we are allowed to look at naturalistic use cases, the results will be much more viscerally usable to humans.
Are there plans to do more long-term research, like if this is sort of something that takes a few exposures to have an effect on your sleep spindles? The next step, what are your next steps?
Yeah, sure. So this is actually a pilot study. So if this goes well, which it is not yet done, but if it all goes well, then I think the plan would be to have a fully funded, much larger study. But I do have to add the caveat that the numbers for our results are, according to like the very, very most senior PI in our lab, unlikely to change a whole lot. So unless I can find like hundreds of thousands of other participants.
I was just wondering, do you compare them to like their original score, or is it like averaged together? Do you compare it to like?
So the numbers that I have are just from the first 2 sleep periods. But we we aren't able to take really any measures. There is a lot of stabilizing of their sleep schedule that occurs via like journaling, and a lot of, like, we ask them to stabilize their sleep schedule and pick a specific window of 8 hours. Did that answer your question?
Yeah. And then how many days have passed when you scored the sleep spindles
Ooh. So,
and why? Yeah.
yeah, okay, so this is something I've learned about sleep science. We generally will consent and then put someone through a sleep lab like one at a time. So these participants can each be studied, like, you know, there can be months in between them. I think that if I'm recalling correctly, the last one prior to me scoring was in May, that the sleep study was finished. So it does take some time sometimes to recruit for some of our more niche studies.
So is it for criteria, it's just like confirming, so the user group, they have to test positive for THC in their system, correct? And then the non-users have to have a clean UA, right?
Yes.
Are you assessing their actual sleep during the night to know, you know, if there is a difference in that between the groups?
I personally am not, but the PSG files do contain enough data that I imagine that you could probably make some conclusions about sleep quality, though the work that I did does not focus on sleep quality. Though sleep time, everybody was in that bed for at least 8 hours or else.
Unlike the rest of us.
Were they allowed to sleep in? Or were they woken up?
I do not think that they were allowed to sleep in. There's a pretty strict timeline in the sleep lab. Depending on the study, there are different pretty specific protocols. And this one had one, I believe, about light. So the light was actually changed at specific times.
I'm sure you said this, and I'm just blanking. How many participants were there?
There were 18 total, and so far I have counted sleep spindles for 8 of them.
Few minutes before we get started with the next one on time. Next slide.
But this one you decided. Like,
You did a fantastic job.
Did I get that?
I was so proud. Yeah, you did really made that.
Yeah. And I always like. And it takes me
Not significant. Yeah, no, same about. No one's really late review, I think. Science, right? Yeah, no, 'cause there's- Yeah.
It's not actually like one place, so. Right, like it's what if like a. Like the one of the reasons why states for it's because it's gonna make you like how would it. But that's what I did do. You know, so. And
Right, So we'll move on from the presentation, and I'll just note it's really exciting to about some of the research that scholars have done at other locations going away for the summer. So the next from Chibuke, who I'm not even going to try to say the title.
Good morning. My name is Shavika. I'm a rising senior at Portland State University majoring in biochem. And this summer I had the privilege to conduct research under Dr. Celestine Wanjala at Vanderbilt University Medical Center in Nashville, Tennessee. The title of my summer project was GPR 56 exerts an interferon beta independent anti-inflammatory effect in CGC CD4 T cells. So a little bit of background information about the project is people with HIV have higher incidences and prevalence of cardiovascular disease and diabetes than HIV negative people. However, this relationship is not explained by the differences in traditional risk factors. It persists despite suppression of plasma viremia on antiretroviral therapy. And chronic inflammation from HIV infection have been linked to accelerated atherosclerosis. So what are CD4 T lymphocytes? Dr. Wanjala's lab created the moniker CGC, which stands for CX3, CR1, GPR56, and CD57. CX3CR1 is a chemokine receptor that traffics cells to inflamed epithelium. GPR56 is an adhesion molecule and also acts as an inhibitor molecule. And then CD57 is a marker of senescence and late activation. The marker combination of CX3, CR1, GPR56, and CD57 on CD4 T cells may reflect antiviral activity. The enrichment of CGC CD4 T cells in the blood and adipose tissue is a feature of HIV infection. And then adipose tissue CGC CD4 T cells have an RNA transcriptome characterized by leukocyte activation and cytotoxic genes. These three surface markers are significant because the marker combination on CD4 T cells may reflect antiviral activity. So they may also be targeted in therapeutics. So that's why we were interested in the marker. So this slide is shows adipose tissue CD4 T cell surface markers. As you can see, the CD57, GPR56, and CX3CR1. And from left to right, we have non-diabetic, pre-diabetic, and diabetic. So CGC CD4 T cell counts increased with progressive insulin resistance in HIV positive individuals. You can see on the flow illustration that the bottom section of the adipose tissue is where these cells would be found. And as you go from left to right, you can see that there is an increase in these cells. So what is GPR56? GPR56 is a member of the adhesion G protein coupled receptor family. Expression of GPR56 is restricted to cytotoxic natural killer cells and T lymphocytes, and increased expression of GPR56 have been linked to greater CD4 T cell pro-inflammatory cytokine expression. So CGC CD4 T cells are associated with a higher risk of cardiovascular disease and metabolic disease in people with HIV. And the specific function of GPR56 and CGC CD4 T cells is undefined. So GPR56 was a marker we used to ID CGC cells, and the fact that this marker seems to be important in cytotoxic T cells leads us to believe that GPR56 is actually a good marker. So we can identify the cells that could potentially be pathogenic with this marker. However, we don't want to knock out GPR56 cells instead. We just want to target pathogenic cells and remove those instead for future therapeutics. All right, so for our hypothesis was that GPR56 has an anti-inflammatory role in CGC CD4 T cells, and a greater expression counteracts a greater CD4 T cell flow inflammatory cytokine expression. To test our hypothesis, we first determined if GPR56 was successfully knocked out from the MOLT4 cell line. Then we compared the CD4 T cell proliferation between wild type and GPR56 knockout. And then lastly, we compared cytokine expression between CD4 T cell wild type and GPR56 knockout. So first we knocked out GPR56. On the left, there's a diagram of a flow diagram that shows that the GPR56 was completely knocked out from the MOLT4 wild type, which is significant because sometimes you have a partial knockout. In this case, we have a complete knockout, which will lead to better results. So we confirmed the GPR56 knockout using flow, and then we stained the antibody against GPR56 in agua. A little bit about the cell line. We used the MOLT4 cell line, which is a CD4 human T cell line expressing GPR56. We use for GPR56 knockout cells to try to understand the role of GPR56 and CD4 T cells, and then using flow, we confirmed the successful knockout of the GPR56 gene. So we cultured the cells in RPI medium supplemented with 10% FBS and sodium bicarbonate, then plated the cells in 96 well plates that I stimulated with CD3 antigen and CD28. And once we plated the cells, we noticed after 48 hours that there was a slight difference in growth. On the left, you have the wild type, and then on the right, you have the GPR56 knockout. On the left, you can see how the on the bottom of the 96 well plate that the cells are more pelleted, while on the GPR56 knockout, it's more of a monolayer. And this trend can continue after another day, so 72 hours. So the next question we had is, does knocking out GPR56 affect the growth of these cells? And to answer this question, we stained the cells to determine whether there was a difference in the proliferation of the cells, because we thought, just by looking at it visually, that there seemed to be more cells in the wild type than compared to the knockout. All right, so we for multiple wild type and GPR56 knockout, we figured out that they actually had a similar growth in culture. So this is what both the cells look like under the microscope. Morphologically, they're similar or identical. And then as well, we conducted a cell trace experiment. So to examine if there was a difference in growth between the knockout and wild type, we stained the cells with cell trace, which is a marker that doesn't leak out. But as the cells divide, the amount of cell trace dilutes. Therefore, with each division, we have half the amount of cell trace from the previous cells. And our results showed after 3 days, there was no difference in cell proliferation. And if there was a difference, we would see another peak, because this is parental, this is P1, so we would see a peak to peak, or just another peak in general, if there was. So knocking out GPR56 led to different growth in culture, but did not reduce the viability. So therefore, this is a valid experimental model, because we did not kill the cells by knocking out GPR56. And then after we confirmed that it was a valid experimental model, we conducted a qPCR cytokine analysis. In a qPCR, RNA is first transcribed into complementary DNA and then reverse transcriptase from total RNA. The cDNA is then used as a template for the qPCR reaction. This method was used to determine the rate at which these genes of interest were transcribed by the CD4 T cells. The two genes we were looking at were interferon beta and then tumor necrosis factor alpha, TNF alpha. And then after we conducted qPCR, this is the results. So GPR56 knockout multiple cell expressed a higher level of TNF alpha. So as you can see on the graph, there's a 14-fold, after 72 hours, there's a 14-fold increase. And in INF beta, we saw a 3-fold increase over 72 hours, which is unexpected because INF beta is a cell regulatory mechanism that prevents inflammations. And since INF beta expression increases, it's expected that it's GPR56 independent. To conclude, no significant difference was found in the viability and proliferation between knockout and wild type of CD4 B cells. Knocking out GPR56 led to increased expression of the inflammatory cytokine TNF alpha, and GPR56 anti-inflammatory activity appears to be interferon beta independent. So for future directions, more research is needed to understand the mechanisms by which GPR56 suppresses pro-inflammatory cytokine transcript expression. Future experiments will examine the relationship between GPR56 and cytokines, and the role of GPR56 in HIV infections will also be studied using the multiple cell lines that we have characterized. And then lastly, GPR56 anti-inflammatory function could be targeted by therapeutic interventions to disrupt CGC, CD4 T cell pro-inflammatory cytokine expression. So for my acknowledgements, I'd like to convey my heartfelt appreciation and gratitude to my SRE mentor, Dr. Wanjala, as well as the administrative team of URISE and Exeter for providing me with the opportunity to work on this wonderful project. I'd also like to thank Dr. John Curtis' lab and Dr. Celestine Wanjala's lab for their assistance completing this project in a short period of time. Thank you. Any questions? Yeah.
I was wondering if you had any idea of what the mechanism behind the GPR56 might be.
Yeah, so it's typically it is a adhesion molecule. So I think I don't know if I asked you specifically, but I did by the mechanism you mean by which it does, like, yeah, I didn't go that deep into it, but I believe like it is it's an adhesion deprotein coupled receptor family. So it technically, I believe it like binds the cells, I guess, or it makes them grouped together. So maybe that's why we saw the results we see with the pelleted compared to the monolayer. So since when we removed the GPR56, we saw how it created the monolayer. So I don't know the mechanisms though. I could look it up and get back to you for sure though.
Thank you.
Yes,
Thanks, presentation, Drew.
thank you.
Can you talk a little bit about it seemed like the interferon and the other one rose at a different rate.
Mhm.
Any idea why? So tumor necrosis factor was a slower rise and the interferon ran even by your second time point, by 40, it's really at, is there any idea about the difference between those two markers?
No, we did have the error. It could have been like just an error on my part, but the error part was pretty large. It could just be the concentration was higher in the mold for GPR56 knockout to begin with. I know during qPCR it's supposed to be normalized, the two cDNA, but it could have just I mean this is my first time in a wet lab, so I may have made a mistake unfortunately, and then while analyzing the data that may be the
Well, your
results.
second point, the data looks really tight.
Yeah. But yeah, as of right now, I can ask Dr. Mundala to maybe go over with me again and then see and get back to you on that. So of course.
So I don't pretend to understand half of that, but I remember with COVID, we heard about these cytokine like that was a big thing for the problems with COVID, right?
Mhm.
So could this have implications in what's happening with COVID?
Not necessarily, I would say. I mean, in a way, because these markers are bound on people's ethos. So it could definitely have implications just because it is an immune cell, but like mainly this is you can see the difference in HIV patients. So unless the patient had COVID and HIV as well, you probably wouldn't see any real crossover implications. So yeah. Yes.
You mentioned it was your first time in a wet lab.
Yeah. So at first I was waiting to get on board, so I had to learn how to like a lot of the basic skills like pipetting, and then after that I was just helping out Dr. Mundala for a little bit while I was trying to understand my project. So, but to begin with, we first for my project specifically, I first started with culturing the cells, like as you saw the multiple cells. So I cultured those and fed those every day or every 5 days, and then once they were dense enough, I plated them personally. So and then I also stimulated the plates with the 96 well plate, and extracted the RNA DNA. I did the whole qPCR process as well. So for my first project, I actually went like hands-on, full straight into it. So like I was really intimidated, especially being at Vanderbilt University. It's like a pretty prestigious university. But like Dr. Mundala was really cautious, and she took her time with me. So which I really admired about this whole process, and being in a wet lab in general. I knew I was going to make mistakes, but then we looked over those mistakes and just continued on. So it was a great experience, I would say. Any other questions?
So you conducted all of this research within a time
Yeah.
of about 2.5 months?
It really actually like about 5 weeks, because I had to get onboarded at Vanderbilt, which took some time as well. Yeah, I think one of the things we can work on for future directions is also just the longevity and longitudinal of the study. That would probably be beneficial to really understand the results. These could definitely change if it was a longer study, but for the short time I was there, this is what we came up with. So yeah.
Here.
Thank you.
Awesome. Hello, everyone. Thank you all for being here. My name is Joy Jimenez, and today I'll be talking about a research project that I had the opportunity to conduct at the University of Iowa this summer, in which I titled Stress, Parent Practices, and Children's External. Aggression and conduct problems. These are some of the behaviors that have increased both in severity and frequency among children and adolescents all around the United States. A meta-analysis including 41 studies from different countries found that around 113 million families are being affected by their children's externalized behaviors or disruptive behaviors. And so studies have tried to find why this is occurring. Why are children acting out like this? Parents are asking themselves too. And there are various factors, psychological, biological, and environmental factors. One of these being stress, particularly stress in your microsystem, so stress in your family environments. And so research has tried to investigate what explains the association between stress and externalized behaviors. And they found that harsh parenting practices, things such as corporal punishment, explain this association. However, little attention has been paid to the other side of the spectrum in terms of parent practices, that being positive parent practices. And this is where my project begins. To gain a more holistic understanding of how parent practices plays a role in the association between stress and externalized behaviors, I decided to look at positive parent practices, and I want to see if they serve as mediators. So I looked at these three variables: parental stress, parent practices, and children's externalized behaviors. But what is parental stress? In my project, I decided to use two categories of stress. I looked at the frequency of distressing events that a parent experiences throughout their life. And these are things such as losing a loved one, being incarcerated. And I also looked at parenting stress. So it's the stress that parents get for just meeting the demands of their children and just being parents, which I want to mention and note that having parenting stress is totally normal and it's part of normal life. And we use the life event scale to measure the amount of stressful life events that parents were experiencing. And we use the parenting events instrument to measure the parenting stress that the parents were experiencing. Now the operationalization of parent practices. I decided to look at warmth, involvement, and autonomy and support. And I have included the various instruments that we use to measure each of these variables. Now, externalized behaviors. I've been talking about externalized behaviors quite often, but what are externalized behaviors? Externalized behaviors are outwardly displayed behaviors that are complicated and disruptive. Things such as hyperactivity, aggression, vandalism. And we use the child behavioral checklist to measure these children's behaviors through that instrument. Now moving on to methods and materials. So we recruited our participants from Iowa City, Iowa, and surrounding areas. We had a total of 144 children and their families participate in the study from ages 3 to 7. And here are the demographics of the participants. 50.7% of children were male, and the majority of the sample had the following characteristics. They were white, their parents were married, and their parents were highly educated, so they had a bachelor's degree or above, and they were also middle or upper class. And I have a graph here with the racial composition of the children in this study. Now procedures. This was a quantitative study, and it was survey-based. We had the parents take 6 different questionnaires, and I want to make a quick note that all the measures, instruments, and data used in this project were part of the longitudinal school readiness study at the University of Iowa. And these are some examples of the stress questionnaires that the parents took. And these are some examples of the parent practices measures. And here is the child behavioral checklist. And here's an example if you guys want to read through that. We used 2 versions of them, one from children's 3 to 5 and the other one from children's 6 to 7. Because they have different questions and they vary depending on the behaviors of the children, of course. And the interesting thing about this questionnaire is that we had multiple raters, which increases reliability, of course. And so we had the primary caregiver. And by primary caregiver, I mean the person who spends most time with the child, guardian, biological or not. And then we had the partner of the primary caregiver, if applicable, so the second parent. And then we also had this secondary caregiver, usually in the form of a teacher or a babysitter, considering the age of the children. Remember that it's from ages 3 to 7. So analysis and results. So these were some of the covariates that I looked at, such as parent sex, family, socioeconomic status, and we ran several linear regressions and 2 multiple mediation analyses using SPSS, which is a social science software that we use to use statistical data. And here are the results. We found that parents who display, who have higher levels of stress usually tend to display less affection and are less involved and are less encouraging towards their children. And we also found that parents with lower levels of stress tend to display more positive parent practice, more involved in their children's life, their school activities, and those children also display less externalized behaviors. So they're displaying less aggression, less defiance. However, contrary to harsh parenting practices, positive parenting practices do not serve as a mediator, but this data does provide more holistic understanding of the role of parent practices. looking at the spectrum, positive and negative. And the implications. But what does this mean? This means that we have to make intervention programs for families and children. And why? Because it reduces future criminalization rates or incarceration rates, which is very important. There's research saying that externalized behavior during childhood can lead to adolescent or adulthood criminal rates, which are increased. And so we need to attend to those externalized behaviors. And the intervention programs that we need to do have to be aimed at reducing parent stress, both in frequency and related to being a parent, and also encouraging positive parent practices and providing families and children with skills. And in conclusion, the research that was presented in this presentation replicates and extends prior research. And however, there was a limitation to the study, which was that we had a relatively similar sample. And so it reduces external validity because we can't generalize these findings, of course. Remember, 61% of the sample was white. And so for implications that I previously mentioned, I would encourage using more P3 programs, which are positive parenting programs, which encourage positive parenting practice among children and encourages the positive attachments between children and their caregivers. And in terms of future research, I would consider further investigating parent-driven mechanisms that can explain the association between stress and externalized behavior, and also changing the conceptualization of stress. We can use different stress measures as well. And in essence, I found that child welfare is family welfare. We can't fully support a child if we're not fully supporting a family. And yeah, I just want to mention again that the study was part of the longitudinal school readiness study. I've provided a QR code if you guys want to learn more about the study, but it is based in Iowa, so you can't really be a participant. And I have also included this QR code for the references that I used for my poster and for this presentation. And I just want to give a huge thank you to the University of Iowa and the Graduate College at the University of Iowa for funding my stay and my research throughout this summer. And also a huge thank you to Dr. Isaac Peterson and Ben Swanson for the guidance and support in this project. And also thank you to URISE and Build Exito for letting me present my presentation here and for introducing me to research. And yeah, these were some of the wonderful people that I've befriended this summer and who I dearly miss. But yeah, thank you so much for listening. Any questions?
So like, did you find that you had low, so okay, I'm assuming the demographic that is open, it's not just local average.
Yeah, it's a community.
So did you find anything interesting like single parents or parents with children with disabilities or anything like that?
That is part of the longitudinal study. However, I just focused on measuring the stress of the primary caregiver. So I wasn't really looking at the family or dyadic relationships in those families. I was mostly focused on the child and the primary caregiver. Yeah, but there is information on a case study that further information on that. And so sorry, I couldn't be like more precise on the specific data, but yeah, the study will focus on that. Yes.
How does you mentioned like the percentage of the participants that were like white or educated, or they were all educated, but how does that sample compare to the population, like in general?
Yeah, oh, okay, well,
Yeah.
so it is very representative of the population in Iowa City, considering that Iowa City is a college campus, so it's very representative and applicable to that city. However, like I mentioned, it is a limitation. We can't really generalize the results because considering somewhere like here in Portland, we're a lot more diverse. So that's something that could be replicated, not replicated, but modified in future research. Yeah, but it does affect in terms of different covariates that we can add and things that we can find. Yeah, sorry.
No, it's okay, it's okay. Were you able to get any data on the actual like the percentage of children who like, whether it was like past data or what you were actually doing, the children that actually went on to have delinquent behaviors?
Yeah, so this is a longitudinal study, but we're only including participants from ages 3 to 7. But there is other research with other ages that, and it was a longitudinal study as well, that we were observing children from ages like 10 through 18, and we did see an increase in those children and families that were not given those intervention programs or were not attending to their children's externalized behavior. So other research does back that statistic up, but I don't have a specific number that I can give to you in terms of this project.
What was like the number one stressor for parents?
I didn't look at the number one stressor. I just looked at frequency and levels of stress in terms of parenting related stress. But a lot of things such as divorce and losing a loved one we noticed were quite prevalent in terms of like the occurrence that we saw amongst primary caregivers. Yeah.
2 questions. One is, were the children controlled for diagnoses like autism, ADHD, OCD, that kind of stuff? And the second question is, did the family have to identify only one person to be the primary caregiver, and if so, why?
Great questions. Yeah. So our RAs cannot give clinical diagnoses. They're just assessors. So that is something that we mentioned in the criteria, and then we mentioned upfront to the participants and their families. We are not giving any diagnoses. We cannot talk about medical providers. Yeah, it's not a screening. It's not like clinical based. It's just for research. Yeah, and because I should mention that our RAs are undergraduates, so they do not have the credentials to even provide diagnoses. And on to the second question, we identify the primary caregiver by asking the families who is the person who spends most time with the child and the person who had those lab visits, because we have 4 lab visits in total. 2 are 1 week apart, and then the other 2 are 9 months afterwards. So we want the same primary caregiver to attend those 4 lab visits. So whoever can attend those and whoever spends time, we classify as a primary caregiver. But there were some instances where there was just one caregiver. There wasn't like a partner of the primary caregiver.
But if there was more than one caregiver, the family had to choose one person.
Yes, that is correct. Yeah.
All right, well, thank you.
Good morning, everyone. I am Dr. Palacios. I'm a psychology undergrad at KMC and also a URI scholar. I am part of Dr. Angela lab at OHSU, and I'm gonna present the effect of metformin on acute and chronic binge-like drinking on inbred high drinking in the body. So a little bit of background about this study is that metformin is an activator of adenosine monophosphate, activated protein kinase, which is also known as AMPK. And AMPK is responsible for regulating cellular energy in many body tissues. Metformin has been shown to increase the phosphorylation of AMPK, which is the activated form, also known as PAMPK. And however, ethanol and cocaine decrease the phosphorylation of AMPK in many brain regions that are important for motivated behaviors. Since metformin has been shown to reduce cocaine-seeking behavior in rats, the purpose of this study is whether it can change alcohol drinking. So for this study, there were 3 experiments. The first one was to determine the effect of metformin on acute binge-like drinking using a binge-like drinking assay, which is drinking in the dark. For experiment 2, it was to determine the effect of metformin on chronic binge-like drinking. And for experiment 3, it was to determine whether metformin changes AMPK phosphorylation in the nucleus accumbens, which is a brain region important for addiction. So for experiment 1, the mice were binge drinking for 3 days, but prior to day 4, they were injected metformin prior to alcohol access. And it was found that metformin reduced ethanol intake, as you can see on the orange bar, and the gray bar is the saline control. So this suggests that metformin may be helpful in reducing excessive drinking at an early stage of abuse. For experiment 2, the mice were drinking ethanol or binge drinking for 4 weeks, 4 days a week. And prior to alcohol access on week 5, they were injected metformin. And it was found that metformin only reduced binge drinking in days 2 and 4. As you can see in the graphs, there wasn't a difference in days 1 and 3. So this suggests that metformin may be helpful in reducing excessive drinking in those with a history of chronic drinking. For experiment 3, I am in the process right now of determining whether metformin changes AMPK phosphorylation in the nucleus accumbens. And for this experiment, the mice were followed the same experiment as experiment 2. But the brains were collected right after the last drinking session, and the AMPK and PAMPK will be measured. And how are we gonna do that? We're gonna use a technique called Western blotting, and we're gonna extract the proteins, and we're gonna measure the amount of proteins, and we're gonna load equal amounts of proteins into a gel, and we're gonna separate them by size, and then we're gonna transfer it to a membrane to and after transferring to the membrane, we're gonna block it, which means that we're gonna use some antibodies to bind to specific proteins that we want to look for, and then we're gonna quantify the proteins to measure how much proteins we have, and those measure the amount of PA and PK. Well, thank you, everyone, your risk coordinators, especially Shandy Dixon and Dr. Kater Foster, and also all of the Osborne Lab members, and also the mice in the veterinary medical unit at the Pro MBA. So now it's time for questions.
Curious that you saw the reductions on day 2 and 4, but not on day 3. So is it like they can go, these heavy drinkers can go a day without, but then they need to get back together? Like, well, why was there a fluctuation across the days?
Yeah, I wasn't part of that experiment, but we know that actually we don't know why that happened. It might have been like maybe a drug effect, maybe the drug has takes some time to make the effect, or it has like that variation. And yeah. Yes.
That's a fascinating study with really interesting results. But I was also curious if the group that received metformin, if it changed how much how much food they consumed as well, if you observed that.
It wasn't measured like the food, but it was quantified the water intake and also like saccharin intake or like reward, but it didn't change water or saccharin intake. It was only the ethanol intake that was affected. It's
really nice to talk about them. As it seems like metformin is a pretty promising, considering that you did the controls to see if it was specific for the ethanol intake. I was wondering if you know that metformin has been used as a drug for any other different types of disorders of disease.
Yeah, so I think that the main use for metformin is for diabetes, and also for cocaine seeking in rats. Other than that, I don't know whether it's been used in other circumstances. So. Yes.
Do you know of studies that have looked at metformin and alcohol in humans, clinical models?
No, I think I have not looked into that.
It's pretty exciting research.
Yeah, it's definitely future directions.
I had a question on the mice. So before testing them, they were already binge drinkers or, and then I'm just curious how they're, I guess, how you measure if they're an increased binge drinker. I think you mentioned, like, are they expected to drink a particular amount of alcohol or something, or is it, they're not given alcohol? Because I know it's in mice, so it's different than humans. I just needed that.
Yeah, so no, they were not drinking before. They were pretty young mice. And the ethanol intake was measured with graduated bottles that were given to the mice to drink like over the sessions. It would for 2 hours or sometimes 4 hours for the second experiment. Yeah, that was how it is measured. Well, I guess yeah.
So AMPK is known as like for energy metabolism. It's highly stimulated following physical activity. So has anyone looked at maybe physical activity as like a way to stimulate AMPK rather than a pharmacological approach? Or do you see that as like an avenue for therapeutics beyond just like, or in combination with that? Like if the AMPK is like a pathway of interest and exercise is known to stimulate the pathway, why not just like get people to be a little more, go for a walk instead of like shooting up? That makes sense.
Yeah, I guess that's a great question. I honestly don't know how to answer that. But yeah, I guess that's something to look for.
I think it's mixed, yeah.
Yeah.
Cool. And then do diabetics, I guess primarily type 2, if they're on metformin, are they less prone to drink? Like is anyone looked at that? I mean, there might be health reasons as to why they're not drinking to begin with, but maybe they don't have as much of a desire to drink because they're on metformin. Have you looked into that, or is that something for consideration?
Or clarify that question.
So like it's a common diabetic medicine. I'm just curious whether an individual with diabetes who's on metformin might just be drinking less because of that drug.
Oh, that's a good question. Well, I guess it might be due to metformin, but I guess that's something to
Let's check literature. Cool. Good talk.
So the last question I had was, so they're drinking less on metformin, but are they still drinking to the point of intoxication? I guess.
yes,
They are.
yeah. Thank you, everyone.
Okay, so again, we've had excellent presentations today, once again, and also research questions generated. So that concludes our presentations, but we still have an hour to go with posters. So if you wanted to follow up with any of our presenters and talk to them more about their research, you please find their posters after we have a few comments in the atrium. So join us in the atrium.